Today we're going to talk about the urinary lab. And so the function of the urinary system is to filter toxins, primarily I should say, is to filter toxins from the blood, and then we remove that, of course, as urine when we urinate. So we're going to start with the kidneys themselves. I have this model here. As you can see, I've removed the abdominal organs here so that you can see the kidneys. They sit here just against the dorsal wall of the body. You'll notice that the right kidney is slightly lower than the left. That's because it gets crowded out by the liver. And you also notice just the top of the kidneys, the gland there. That's the adrenal gland. We talked about that. And so the job of the kidneys, filter the blood. We know that the renal arteries deliver that blood to the kidneys. And then the kidneys filter. Any filtration is moved out of the body through the ureters, stored in the bladder, the urinary bladder, and then they leave the body through the urethra. So in the case of the kidneys, they get their name based on their shape. So this would represent the shape of the kidney. The right kidney will draw slightly lower, since it is crowded out by the liver. The kidney itself is found in a space called the retroperitoneal space. Retroperitoneal means that it's behind the dorsal tube, the peritoneum. The peritoneum is the serous membrane that covers the abdominal organs, and the kidneys lie posterior to that in a space called the retroperitoneal space. Just the top of the kidneys, we have the adrenal glands. We know the adrenal glands, part of the endocrine system, secrete hormone from the adrenal cortex as well as the adrenal medulla. So we'll just draw those here today, but not discuss anything about the adrenal glands. They just sit atop the kidneys, kind of like a little hat. The lateral surface of the kidney is convex. The medial surface is concave. This medial surface is going to have this concavity because this is where essentially all the vessels will enter and leave the kidney. The function of the kidney. So the kidneys have several functions. They include regulate total volume in the body. And we talked about that when we talked about the regulation of blood pressure and that long-term regulation by the kidneys, and the kidneys can increase or decrease the amount of urine that's produced. So that's one of their functions. They also regulate the ion concentration in the extracellular fluid. That again has to do with their ability to filter out ions, and then those ions will diffuse from where there's more to where there's less, and that regulates then the fluid around our cells. It also ensures long-term acid-base homeostasis. This is something we'll discuss in the weeks to come, but fluid balance and acid-base homeostasis are also the job of the kidneys. And so the kidneys can regulate how much hydrogen, how many hydrogen ions are in the solutions of the body by allowing for us to excrete more or retain more of those hydrogen ions. Our kidneys also have the function of excreting metabolic waste, toxins, and drugs. So that means part of their filtration of the blood is metabolic waste. Those include things like urea and uric acid that are produced by the cells as waste material, as well as any drugs that we've ingested or toxins we might have come into contact with. The kidneys produce erythropoietin. We've seen that in the process of making erythrocytes. We know that when there is decreased oxygen carrying capacity, then the kidneys release erythropoietin. Erythropoietin then goes to act on the hematopoietic stem cells in the bone marrow and stimulate the production of erythrocytes. Our kidneys can also activate vitamin D, as well as in extreme conditions carry out gluconeogenesis, which is the formation of new glucose. Now to talk about the structure of the kidney. So the outermost layer of the kidney is called the renal fascia. We'll just say that that's the brown portion that's drawn here. This renal fascia is connective tissue covering around the kidney. Just deep to the renal fascia is going to be the perirenal fat capsule. So I'll draw that in here. And that just means that there's fat all around the kidney, and that fat there is protected. Often hear, let's say athletes, particularly male athletes sometimes, talk about taking kidney shots, and that's because the kidneys are just there on the dorsal surface of the body. And so to assist in protecting those, we have this fat covering, that's the perirenal fat capsule. And then there is the fibrous capsule. The fibrous capsule will be the connective tissue layer that is going to enclose the tissues that are the tissues of the kidney. So that the fibrous capsule here is kind of the external limit of the kidneys. And so the kidneys themselves have these 3 layers on the exterior to protect the kidneys, because they are they have very little protection there on the dorsal body surface, and they're in that retroperitoneal space. Now the actual structure of the kidney. So these coverings are there. On the outer surface, we have the cortex. So this area that I'm going to draw here in green is the cortex. The cortex is the outer layer of the kidney. And so the job of the cortex is to filter. It looks granular in nature because its job is to be the major site of filtration. Just deep to that cortex, we're going to have the renal medulla. And the renal medulla is characterized with pyramids. So I'm going to draw these pyramids coming in like so. That is going to be the medulla. The job of the medulla is to fine-tune the urine that we're making. So all that means is some of the things that get filtered out are going to, in fact, want to be removed from the body, and other things we don't necessarily want to remove. And so the medulla is characterized by these renal pyramids, also called medullary pyramids. The tip of these medullary pyramids are called the papillae. And the papillae are where urine is going to be released from the kidney. So what gets released from the papillae is urine. We also have in the renal medulla, between the pyramids, the renal columns, and the renal column and the pyramid together make up the lobes of the kidney. And then the innermost portion, or the most medial aspect of the kidney, is going to be the renal pelvis. And so the renal pelvis begins here at the renal papillae. There's a structure here called the minor calyces. These minor calyces then merge with each other to form the major calyces. So here we'll show the junction of 2 minor calyces into a larger structure, which would be the major calyces. And so the pyramids empty into the minor calyces. The minor calyces merge into a larger structure that is the major calyces, and then all of the majors merge into the renal pelvis. From the renal pelvis, there's going to be a tube that leaves the kidney. These tubes, from the minor calyces, major calyces, renal pelvis, we have this tube from each kidney. These are called the ureters, and their jobs are to transport urine to the urinary bladder. And so urine is going to be initiated there at the papillae, travel through the minor calyces, then the major calyces, the renal pelvis, and then the ureter. There's an extensive blood supply to the kidneys. So we're going to lay on top of this image here the abdominal aorta. You'll remember that the abdominal aorta has 2 large branches that are going to come off. Those are going to be our renal arteries. And so that would be left then right renal artery. They are large because at any given time, these kidneys are receiving between 20 to 25% of your total oxygenated blood. So that means the kidneys themselves are, the two of them, are receiving a significant amount of blood for filtration. It's very important because this is where we're going to cleanse that blood of metabolic waste. And so the renal arteries are going to branch. Once they get here into the kidneys, and the goal is for them to continually branch. So the first branch of the renal artery is going to be the segmental artery. And so the renal arteries branch into the segmental arteries, then the segmental arteries into the interlobar arteries, the interlobar arteries into the arcuates, and then the arcuate into the cortical radiate. And the cortical radiate are going to end up here in the cortex portion of the kidney. So again, we have the renal arteries coming directly off of the abdominal aorta. The renal arteries are going to branch into the segmental arteries into the interlobar. You can imagine that's between the lobes that we talked about, the columns that are joined, medullary pyramid make a lobe. The interlobar arteries branch into the arcuate, and the arcuate arteries into the cortical radiate. The cortical radiate artery is going to branch then into a capillary. So the cortical radiate arterioles will lead into the capillary bed. And so that arteriole is called the afferent arteriole. It will then lead into the glomerular, we'll just call it the glomerulus. Then we have the efferent arteriole, peritubular capillary, we'll put here in parentheses that this is also a capillary. So what happens in the kidney here is that we have those two capillary beds, and those two capillary beds here are going to allow for sufficient filtration. That means all the waste material gets removed from the body. And then from the peritubular capillary, we're going to have the cortical radiate vein. That's going to then drain, of course, into the arcuate vein. From there, we're going to have the interlobar vein. There's no segmental. And that will empty into the renal vein. So that's our blood supply through the kidney. So the blood comes in through the renal artery, they branch into segmental, the segmentals branch into interlobar into arcuate, and then the arcuate branch into cortical radiate. Remember, this was the cortex. And so the cortical radiate will then branch into the afferent arteriole that feeds the first capillary bed, the glomerulus, and then from the glomerulus there is the efferent arteriole that's going to feed the second capillary bed, which are called the peritubular capillaries. Those empty into the cortical radiate vein, and then the arcuate vein, and then the interlobar vein, and then out through the renal vein, of course, to the inferior vena cava, and then that blood will be returned. And the blood is going to be filtered and cleansed through these capillaries. Capillaries, remember, are sites of exchange, and in this case, they're going to be allowing for waste to leave the bloodstream. Now we have to talk about the structural and functional unit of the kidney. It's called the nephron. And so the idea is that each one of these kidneys contain 1 million nephrons. I'm going to draw the first nephron here, and then we'll talk about its structure. So imagine that this is our nephron. We have in the cortex here a ball-like structure. That ball has tubes attached to it, like so. And so in the cortex here, we have 1 million of these ball-like structures with tubes attached to them. Those are going to be our nephrons. And so the structure of the nephron is that it has a ball-like structure, like so. And then it has tubes that attach to the structure. So even though I'm drawing it somewhat larger here, you're going to imagine that they are microscopic. And they are found primarily in the cortex portion of the kidney. So this is the nephron. The nephron is made up of 2 parts. They are this portion, which is called the renal corpuscle, and this portion, which is called the renal tubule. And so remember, the function of the kidney is to filter our blood. We have 1 million of these structures, which are called the nephron. This entire structure here, we're running out of space, but renal corpuscle plus the renal tubule are the nephron. That is the structural unit, functional unit of the kidney. 1 million in each kidney that allow for filtration. And so what happens here is at the renal corpuscle, there is the glomerular capsule and the glomerulus. And so the glomerulus branched off of the afferent arteriole. So for our purposes, this is going to be the afferent arteriole. Same afferent arteriole that we talked about here, a branch of the cortical radiate. That afferent arteriole is going to give off a branch here. So it goes like so. But it gave off a branch here. The branch of that afferent arteriole is the glomerulus. So the glomerulus is going to be found here. And it loosely translated means a ball. So that's the glomerulus. The job of the glomerulus is to allow for things to be pushed out of the capillary. So what happens here at the glomerulus is that most substances get pushed across that capillary membrane, across the membrane of the glomerular capsule, and we're going to then have inside of the nephron, the loops of the nephron, most of the components that are inside of the blood, with the exception of proteins and cells. So you'll remember in our plasma, all those components that we have include water, include electrolytes. We also have nutrients. We have hormones. And so everything except proteins and cells get pushed across here at the glomerulus. That process is going to produce a solution that is called filtrate. Once those things get pushed across this membrane, so this membrane is made up of simple squamous epithelial cells and cells that are called podocytes. They have feet, and the feet are designed to catch any large molecule like cells and proteins, so that in the tubule, all that's left are those components of the plasma that are not cells and that are not protein. This process of pushing things across that glomerular membrane is happening constantly, 24/7 in our body, such that in a given day, 180 liters of liquid is going to be processed. So we know that we only have, on average, 5 liters of blood in our body, but our kidneys are able to process 180 liters a day. So that then lets us know that several times over that blood is being filtered, and this process is constantly occurring. Once that filtrate is pushed across, it is called filtrate. From there, we're going to have the glomerulus form its efferent arteriole. So the efferent arteriole is leaving here. That efferent arteriole is then going to feed into a peritubular capillary. So we'll draw our peritubular capillary here. It gets its name because it's going to surround the tube portion of the renal tube. So this is our peritubular capillary here. Trying to draw the blood vessels in this green color, but also help us to appreciate. So the blood that was in the glomerulus leaves this efferent arteriole, travels around the nephron and what is called the peritubular capillary. And then eventually this peritubular capillary is going to merge into a larger vessel. That larger vessel is going to be the cortical radiate vein. And this is the cortical radiate vein here that's going to then merge and drain into the arcuate veins. They will merge into the interlumbar, and then the renal vein will escape. So we've said that this is the glomerular capsule. There's the glomerulus. Now we have the renal tube. The renal tube has several parts. It has this tube, which is called the proximal convoluted tubule. I'm going to abbreviate it there, PCT. That's proximal convoluted tubule. This is where we're going to receive all of those components that are pushed across that glomerular membrane. That solution will then, after it got pushed across here, will move. So let's say the solution is being pushed across here. It will move through the proximal convoluted tubule and then inferiorly to the descending limb. This is the descending limb of the nephron loop, so the solutions are moving down. And then those solutions move up the ascending limb of the nephron loop into this structure. This structure is called the distal convoluted tubule. And the distal convoluted tubule is going to empty into this structure, which is called the collecting duct. The idea here is we've pushed this filtrate across at the glomerulus over the membrane of the glomerular capsule, but we have things in here that the body wants. So there's water that got pushed across, there are electrolytes, things like sodium and potassium and calcium. There are also nutrients and hormones, and some of those things we want in the blood. And so this process of having the renal tubule of the nephron adjacent to the peritubular capillaries allows for things to move across the membrane. Right? So things can move back and forth until we have the correct composition in the blood, and then all others will be excreted, and what leaves this collecting duct is then going to leave. This collecting duct is going to empty into the renal papillae. That's the structure that we saw just here. And at that point we have urine. And that urine leaves the renal papillae, goes through the minor calyces, the major calyces, the renal pelvis, and then the ureters out to be stored in the urinary bladder. But we have pushed everything out here at the renal corpuscle and then modified by moving things across. The initial movement of things there at the glomerular capsule is going to be called glomerular filtration. So this process of moving things across, I'm going to put a 1 here. That's called glomerular filtration. That means everything gets moved out of the capillaries into the nephron to be filtered. Then we have tubular reabsorption. I'm going to put a 2 here for tubular reabsorption. These are substances that are moving from the nephron back into the capillary. And so you can imagine it's going to be things like the electrolytes that we want to keep. We also want those nutrients, things like glucose and amino acids, we need those, as well as hormones. That's tubular reabsorption. And then the third portion is tubular secretion. In the case of tubular secretion, these are things that were not filtered out of the capillary initially, but things that we don't want in the blood, and they get filtered out. So I'm going to put a 3 here, showing that we're going from the capillary into the nephron loop, because tubular secretion are things that we don't want. In most cases, these are going to be the drugs that we've ingested that the kidney has the job of removing, and so that's going to occur there. Once we've made the urine, so we start here with the glomerulus, that capillary pushing the substances out. It's a process called glomerular filtration. What is produced here is called filtrate. It includes everything that was in the blood except for proteins and whole cells. Those chemicals move through this proximal convoluted tubule, where we are going to have this process called tubular reabsorption occur. All those things that we want get moved back into the bloodstream. We move along the nephron, the descending limb, the ascending limb. This process of tubular secretion is going to be occurring, where there are things in the blood that we don't want, so those will get pumped into the nephron. And once we get to the end of the nephron loop here, this distal convoluted tubule will empty into the collecting ducts. The collecting ducts empty at the renal papillae. At that point, we have urine. And so during this entire process of moving liquids and solids back and forth, the solution that is inside of the nephron is called filtrate. At the end of the renal papillae, we have urine. That urine travels from the renal papillae through the minor calyces, the major calyces, the renal pelvis, and then out through the ureters. These ureters have the job of transporting this urine to the urinary bladder. So we're going to draw the bladder here. The ureters generally come around like so, and then empty into the bladder. So we'll draw the bladder here, like so. Just imagine that it's empty. That's why it's so small. The ureters themselves are muscular tubes. They also lie in the retroperitoneal space. That means they are posterior to the organs of the abdominal cavity, and they lie outside of that serous membrane. The ureters, muscular tubes with 3 layers. The innermost layer is called the mucosa. The mucosa has transitional epithelium. Transitional epithelium stretches in the presence of urine. And so when there's urine, being moved from the kidneys down to the ureters, which you can imagine is a process that's occurring constantly, then those ureters have the ability to stretch and recoil. The intermediate layer of the ureter is called the muscularis layer. The muscularis layer is made of smooth muscle. Of course, smooth muscle can stretch and recoil, but here it's going to allow for periostolysis. Periostolysis, 2 layers of smooth muscle, and those 2 layers of smooth muscle act in opposition. So we'll pretend that my hands are the 2 layers of smooth muscle. One layer contracts, the other layer contracts. So contraction and relaxation and alternation to move things through that muscular tube. And the outermost layer is fibrous connective tissue. It's called the adventasia. It attaches the ureters to the posterior or ventral wall of the body. These ureters are going to empty into the urinary bladder. And so again here I've drawn this urinary bladder quite small. We'll just assume that it's empty. The urinary bladder is a muscular sac. Its job is to be able to store urine. And so you can see I've drawn it quite small here. In our body, it can stretch more than 2 times its original size and rest in that position so that we can hold in a large amount of urine before having that impulse to urinate. The urinary bladder, also made of 3 layers. The innermost layer is the mucosa layer. It's made of transitional epithelial cells. There are many layers of cells that have a rounded or dome top that will stretch in the presence of urine. And so when we have the urine present there between voiding, that transitional epithelial tissue is going to stretch. The urinary bladder is unique in that it has 3 layers of smooth muscle. And so the ureters have 2 layers of smooth muscle, and most of the hollow organs in our body have 2 layers of smooth muscle, a longitudinal layer and a circular layer. In the case of the urinary bladder, it contains 3 layers of smooth muscle. And this is significant because those 3 layers of smooth muscle allow for additional protection of the bladder. That means it's less likely to burst in the presence of excessive urine because it has an additional layer of force. The outermost layer of the urinary bladder is the fibrous avantasia, and that's connective tissue that attaches it to the posterior or ventral wall. In the male, the prostate gland is just inferior to the bladder, and in the female, it is going to be anterior to the vagina. The urinary bladder is going to have a triangular structure, and so we'll draw it here. I'll try to draw the triangle here. It's called a trigone. It is because the ureters dip down and enter on the posterior and inferior surface of the bladder, and then the ureter is the muscular tube that allows for the urine to escape. And so it creates this triangular structure. It's called a trigone. It's significant because this is where urinary bladder infections are most commonly going to be found, because most of those bladder infections are going to be the result of some bacterium entering through the urethra and then landing there in the bladder. Now to the urethra. The urethra is this muscular tube. This muscular tube's job is to connect the urinary bladder to the external environment so that we are able to void when needed. The urethra is also going to contain transitional epithelium, but only half of it. So let's say this is our urethra here. The half that is closest to the superior half, I should say, of the urethra, that's closest to the bladder is made of transitional epithelium. But as we get close to the exterior, that tissue is going to change to stratified squamous because there's the potential to come into contact with something that might be abrasive, and so it's going to protect against abrasion. Here in the urethra, we're going to have 2 sphincters. We have an internal urethral sphincter and an external urethral sphincter. The internal urethral sphincter is going to be made up of smooth muscle, and the external urethral sphincter is made up of skeletal muscle. And so you can think of these sphincters as being closed except when we are urinating. So they are muscular, they surround the tube, they close, and when they close, that prevents the tube from leaking urine. So these sphincters close, and when we're ready to void, we open the sphincters. Internal, external have to open simultaneously to allow for voiding to occur. In the case of the female, the urethra itself is quite small, between 3 and 4 centimeters, and the male is much longer. So we'll say that this is the female urethra here. Here's the urinary bladder. We'll say of a male. And in this case, the male urethra is much longer. It actually has 3 parts. The male urethra has 3 parts. It's about 19 centimeters versus the female, which is about 4 centimeters. In the male, the urethra is going to have the prostatic urethra, and so the prostate gland sits about here, and the prostatic urethra is that part that runs through the prostate. There's then the intermediate portion of the urethra, and then lastly, there's the spongy portion of the urethra, and the spongy portion of the urethra moves through the penis. So you can see it kind of drawn here. Okay, just imagine, okay? And so that would be the spongy urethra there. So in the male, the urethra is much longer, but they have essentially the same characteristics, where there is internal and external urethral sphincter. Now to urine. So we talked about the blood flow, the structure of the kidney, the function of the nephron, and kind of a summary and a general overview as to the way that we produce urine. Now the urine itself. So urine is going to vary based on things like your diet, but there are some standards, and that is that almost 95% of your urine is going to be water. The other 5% is solutes. Solutes are the solid particles that are dissolved in the solution. The solutes that are present there include these things that got filtered out that the body didn't need. And so one of the major components of urine is going to be urea and uric acid. Those two things are metabolic waste. They essentially are going to be toxins to the body if they accumulate. So they are a given that will always be present in the urine. You also have in the urine the possibility to have electrolytes. And so the electrolytes would include sodium and calcium and potassium, and that's going to depend on your homeostasis. So once your body has sufficient amounts, the rest of it will be excreted from the body. The urine is also going to include creatinine. This is going to be from the breakdown of creatine phosphate, and that's when muscles metabolize. You're also going to have in the kidneys some bile pigments might be present there. In that case, it could indicate pathology, or if they're white blood cells or proteins, that can indicate that there's a problem. The color can vary widely, and so a pale color generally indicates that the urine is more dilute versus a more intense color, which would indicate that the urine is more concentrated, meaning that it has more solutes. The color can range from yellow to pink color. Pink might be the result of something like having eaten beets. The odor also varies, and so it should be slightly aromatic, but of course that can also depend on your diet. In this case, I think about asparagus. It should not have a pungent odor. It does if it has been exposed to the environment for any given amount of time, because the bacteria feed on the waste, and then the waste material from the bacteria give that smell of ammonia when the urea gets broken down. The pH can also vary widely from 4.5 to a pH of 8, again dependent on your diet, but the average is about 6. So it depends on diet. With an alkaline diet like that of a vegetarian, increasing the pH, and an acidic diet, let's say of a heavy meat eater, might decrease the pH. The specific gravity. This is a way to measure the number of solutes in the urine. And so it's done using a spectrometer where the light is passed through. On average, it should be from 1.001 to 1.035. That just means the number of solute particles in the urine. There are some homeostatic imbalances, so things that should never be present in the urine. They include glucose. And so glucose, which may get pushed across here as a nutrient, but all of that glucose should be reabsorbed in tubular reabsorption. If it's not, and it makes its way to the urine, it could indicate homeostasis, something like diabetes, where your body has an excessive amount of sugar in the blood. And so instead of taking it back up, it's excreting it to try to maintain homeostasis. You might also have proteins present in the urine. Again, we talked about here at the filtrate, when we filter, proteins and whole cells should not come across. And so if that is the case, it might indicate that there is some damage to the glomerular membrane here that filters, or could simply mean that there's a homeostatic imbalance where the diet has so much meat in it